Hello students, I am Dr. Geetu Gambhir. Today I bring you the next learning episode in BSc Forensic Science on an important unit on the paper titled Technological Methods in Forensic Science, that is Atomic Emission Spectroscopy. In this lecture, you will be studying about the atomic emission spectroscopy, its principle and instrumentation, wherein you will be studying about the sampling devices and sources, microprobes, spectrometers, detectors and mass spectrometers. Dear students, you will also be studying about the instrumental configurations for the multi-elemental analysis and applications of AES. Lastly, we will wind up the module with the conclusion. Recall that in the last lecture, we studied about the atomic absorption spectrometry technique, its principle, methodology, instrumentation and applications. Let's start our today's lecture with a brief look at what we will be learning today. In the module 1, we will be talking about the introduction, followed by the principle of atomic emission spectroscopy, instrumentation of atomic emission spectrophotometer, and the module 4 with the instrumental configurations for the multi-element analysis, applications of atomic emission spectrophotometry, and finally the conclusion. Atomic emission spectroscopy, also designated as AES, has long been standard method for metal analysis. Recent developments in non-combustion plasma sources have expanded the number of applications and generated a new interest in AES. In atomic emission spectroscopy, a minute part of the sample is vaporized and thermally excited to the point of atomic emission. The energy required for these processes is supplied by an electric arc or spark, or more recently by a laser or plasma composed of inert gas. It is convenient to distinguish these sources from the chemical flame sources used to obtain emission spectra. The atomic spectrum emitted by a sample is used to determine its elemental composition. The wavelength at which the intensity measurement is made identifies the element, whereas the intensity of the emitted radiation quantifies its concentration. In atomic emission spectroscopy, the chemical analysis is carried out by measuring the intensity of light emitted from the flame, plasma, arc or spark at a specific wavelength by determining the quantity of an element present in the sample being analyzed. The identity of the element is determined by the help of wavelength of the atomic spectral line, whereas the intensity of the emitted light is proportional to the number of atoms of the element. Instrumentation for AES may be divided into three major components. Firstly, the sampling device and source, the spectrophotometer and the detector and the readout device. The sampling device and source depends on the type of sample and the analytical data desired. As the emission spectrum emerges from the source, it is focused onto the spectrophotometer's entrance slit, where it is dispersed into its component wavelengths. The optical elements permit the site selection within the source's discharge and thus eliminates the need to move the entire source assembly. The use of mirrors properly placed also eliminates the chromatic and the spherical aberrations encountered with the lens optics. At the exit slits of the spectrometer, the radiation is sensed by a photodetector. In older instruments, photographic film was used to record the spectra, but most modern systems use photomultiplied tubes or diode arrays linked directly to the computer driven data processing systems as depicted here below in the given block ray diagram. Instrumentation of atomic emission spectroscopy. Dear students, let us now discuss about the major instrumental components of AES technique one by one. Firstly, the sampling devices and sources. No single source is best for all applications. In general, for solid samples, arc excitation is more sensitive, while spark sources are more stable. Plasma sources are the best choice for the solutions and for gaseous samples. Their sensitivity enables the trace analysis to be carried out at the parts per billion level. Electrical discharges. Electrical discharges can be used as excitation sources. When the discharge strikes the sample surface, it produces high current densities that volatilize the certain small amount of sample. Atoms in the resulting vapor are then excited by the collision in the discharge plasma. Direct current arcs. The simplest electrical discharge is the DC arc between two solid electrodes. Typically, one electrode supports the sample while the other is the counter electrode. The DC arc consists of high current of 5 to 30 ampere, a low voltage of 10 to 25 volt and discharge usually operating in an ear. Arc temperatures range from 4000 to 6000 Kelvin. Excitation of the sample atoms by a DC arc is thus both thermal and electrical in origin. The resulting plasma of high velocity ion, electrons and atoms produces the atomic emission spectra. The DC arc as the excitation source is commonly selected for qualitative survey analysis of trace and major elements, metals, alloys, plastics, rocks, minerals, soils and various biological materials such as tissue, bone and fluids. An internal standard can be used to minimize the effects of arc instability and the sample matrix. Alternating current arc. Alternating currents can be used to produce an arc. AC arc are operated in either high voltage of 2200 to 4400 volts or low voltage of 100 to 400 volt regions. In both the situations, the arc ceases at the end of each half cycle as the applied voltage drops below the value needed to maintain it. High voltage alternating current sparks. The high voltage AC spark discharge provides the greatest precision and stability of all the electrical discharge sources. It is the method of choice for atomic emission spectrometry, analysis of ferrous metals in the industrial operations. Sparks contain high peak currents and power density, which results in the population of high energy electronic levels of atoms and also more extensive ionization than found in the arcs. Microprobes. The laser microprobe is well suited to the analysis of very small samples or tiny localized areas on larger samples. An optical pulsed ruby laser is focused via a conventional microscope onto a minute area of the sample after the visual focusing has been used to select the sample area. The intense heat of the laser vaporizes the small amount of the sample, leaving the hemispherical crater of about 50 micrometer in diameter on the surface of the sample and produces plasma. Once formed, the plasma absorbs the radiation from the laser beam, thus stopping the further vaporization at the sample surface. Although emission spectra suitable for analysis can be produced directly with the laser sources, cross excitation with the spark discharge results in the improved resolution of the spectral lines and to increased signal to noise ratio. The vapor plume generated by the laser is passed between two closely spaced electrodes that are charged to a high voltage. The electrode gap breaks down, producing a spectrum similar to that produced by the spark source. The laser micro furnishes a tool for examining the interior of the individual cells and can even be used with the living organisms, since it is essentially a non-destructive method because of the small amount of sample consumed. It is also useful in analyzing the inclusion areas of alloys or corrosion spots on the metal surface by those interested in these conditions. Resolution of the laser beam is currently limited to 50 micrometers by the lenses used to focus the laser beam. Non-combustion plasma sources. Non-combustion sources improve the accuracy, sensitivity and precision for the large number of elements over that found with the electrical discharge sources. Handling liquid and gaseous samples is much easier with these plasma flame sources. The higher temperatures and cleaner chemical environments of the plasmas overcome two problems associated with the combustion flames: lower temperatures and the reactive chemical environments. Plasmas retain the convenience of liquid sample handling and the precision of excitation conditions of the characteristic of flame sources. Inductively coupled plasma sources. The inductively coupled argon plasma ICAP or ICP torch is a special type of plasma that derives its sustaining power by induction from a high frequency magnetic field. Initially, argon gas passes through a 25 mm quartz tube and upon emerging at the tip is surrounded by an induction coil. An AC current flows through this coil at the frequency of about 30 megahertz and power levels of about 2 kilowatts. The argon gas stream, the support gas that enters the coil, is initially seeded with the free electrons from Tesla discharge coil. These seed electrons quickly interact with the magnetic field of the coil and gain sufficient energy to ionize the argon atoms by collisional excitation. Cations and electrons generated by the initial Tesla spark are accelerated from the tip of the torch. Reversal of the direction of the current in the induction coil reverses the direction of the magnetic field applied to the mixture of atoms, ions and electrons. The fast moving cations and the electrons, known as eddy current, collide with more argon atoms to produce further ionization and intense thermal energy. A flame shaped plasma forms near the top of the torch. The temperature in the plasma ranges from 6000 to 10,000 Kelvin. Direct current plasma sources. In direct current plasma sources, that is DCP sources, a high velocity inert gas produces high temperature plasma and separates the excitation region from the analytical observation zone. In typical DC arc discharge, the current density and the energy available for atomic excitation are independent of the arc current. If the current is increased, the cross-sectional area of the current path increases proportionally with no increase in the arc temperature. The high velocity gas used to reduce the size of the plasma is also useful in separating the current channel from the observation zone. Introduction of liquid samples into the plasma sources. Liquid sample introduction is equally important in plasma methods. In all cases, the analysis can be only as good as sample introduction. However, optimum conditions for the sample introduction into the plasma sources differ markedly from those for the flame atomic absorption sources. Hollow-cathode discharge lamps as emission sources. The hollow-cathode lamp consists of two coaxial cylinders. The inner graphite cylinder is the cathode, contains the sample material. The discharge material is helium. The radiation is emitted from the negative glow, which is confined to the cathode activity. These samples are often used to determine the elements with low boiling points in high melting point matrices. The glow discharge lamp is similar to the hollow cathode discharge lamp in differing only in the position of the sample. The sample is not placed in the middle of the lamp, but rather becomes one of the outer surfaces that see the lamp. Atoms are released from the sample surface by cathode sputtering. Atomic emission spectrometers. In emission spectrometers, the sensitivity is limited by noise originating from the two major components, the flicker of the source and the dark current of the photomultiplier detector. Since the dark current noise remains constant, it is important to decrease the source noise of the spectral background by decreasing the signal band pass until either the detector noise predominates or the spectral line width is reached. High resolution, high luminosity, monochromators are necessary to isolate a spectral line from its background without the loss of radiant power. Both concave and plane diffraction gratings are used as dispersive elements. A concave grating has focusing as well as dispersing capabilities. It requires no additional optical components, whereas the plane grating requires one or more lenses or mirrors to focus the image slit into the focal plane. Another optical device is the Echelle grating system, which is capable of resolution and dispersion magnitude of an greater than conventional grating of equal focal length. Concave grating instruments. In this configuration, also known as Paskin-Runge mounting, the entrance slit, grating and focal plane lie on the circumference of the Rowland circle. The grating is the only optical component between the entrance and the exit slits. For most determinations, only one array of slits is used. Plane grating instruments. Plane gratings serves only as the dispersing element, and therefore, a pair of concave mirrors is usually required to image the entrance slit onto the focal plane. Different spectral regions are focused on the camera detectors film or different wavelengths on the exit slit by rotating the grating. Echelle spectrometers. Echelle gratings provide excellent dispersion and resolution over a wide range of wavelengths in the relatively compact instrument. The different orders appear in horizontal lines, with the longer wavelengths of the lowest order appearing at the bottom and the highest order at the top. Here, the prism cross dispersing element separates the wavelengths in vertical direction, while the Echelle grating separates them horizontally. Detectors and readout devices. Detectors used in emission instrument systems fall into two categories: photographic emulsions and photoelectric transducers. Most recently, mass spectrometers are being used as detectors for ICP sources. Emulsions have the advantage of integrating impinging radiations over the entire time of the exposure and recording all spectral features simultaneously over wide range of wavelengths. These characteristics allow the weak spectral lines to be detected by using a long exposure times, and they also provide a relatively inexpensive means of obtaining permanent records of the emission spectra. Photographic detectors. Photographic materials consist of a light sensitive emulsions coated on a glass plate or a plastic film. The emulsion contains light sensitive crystal of silver halides suspended in gelatin. On exposure, the silver halide crystal that receives radiation from the latent image, subsequent chemical treatment converts the exposed silver halide crystal into a black deposit of silver at the site of the latent image. After development, the emulsion is fixed in a solution that dissolves the unexposed silver halides. Finally, the photographic material is washed thoroughly to remove the chemicals used in developing and fixing. The entire series of operations follows rigidly controlled conditions with respect to time, temperature and chemicals. Photoelectric detectors. The photomultiplier tube and the junction photodiode are the two transducers used almost exclusively in the current AES instrument systems. Photodiodes are used in applications where the detection of lower radiant power is not required. The exit slit of the monochromator must isolate radiations from the individual lines by precise positioning of the slit and accurate control of the slit width. Alternatively, a more complicated mirror system within the spectrometer may be used to subsequently focus radiation on the varying wavelengths onto the exit slit. The radiant power of the spectral line that impinges on the slit is converted by the PM tube into the current that is then used to charge the capacitor resistor circuit. Two modes of integration are used to average the PM signal, both with integration time of 25 to 40 seconds. In the older method, the capacitor voltage is the quantity which read out. The voltage across the capacitor at the end of the sampling time is a function of accumulated charge. Thus, the capacitor voltage is proportional to the time integral of the radiant power. The powers are now compared with the operational amplifier circuits that output the voltage ratio from two lines. In integration with constant time, a voltage to frequency circuit is used to convert the capacitor voltage into electrical pulses of equal height. By counting the pulses generated during the integration period, a number proportional to the radiant power is obtained. This measuring technique has the advantages of large dynamic range extending over 6 decades and built-in noise reduction. The output is usually in digital form, ready for computer processing. Direct reading computerized spectrometers are usually calibrated with the high and low concentration standard. Mass spectrometers. The mass spectrometer systems used for the purpose can both process data and generate reports. The detection limits for the many elements are significantly lower than those observed for the ICP AES systems with the photographic or photoelectric detectors. ICP MS systems also permit the determination of the isotopic composition of the samples. Detection systems used for multi-element analysis are either sequential or simultaneous. In the former system, the dispersed radiation is transmitted sequentially to the single detector. This is generally accomplished by scanning monochromators or rotating filters. Scanning monochromators have a grating or mirror usually controlled by a computer driven stepper motor. The most common simultaneous or parallel systems are multi-channel with one or more detectors for each analyte element. The multiple PM tubes positioned on the circumference of a roll-in circle, the PM multi-channel module used with an Echelle spectrometer and linear photodiode arrays are examples of simultaneous detection systems. Multi-element simultaneous analysis is used effectively in situations that require routine determinations. Instruments used in such analysis, sometimes called as quantometers, having widespread applications, especially in the ferrous metal industries. For less routine analysis involving greater control of optical parameters, the instruments with greater flexibility are required. If sample sizes are adequate, sequential multi-element instruments provide the flexibility needed. The sequential system shown employs a double monochromator to minimize interference from stray radiation. Computer controlled stepper motors provide fast movement of the grating to the desired wavelength. The plasma plume observation height is optimized automatically for each element by moving mirror M1 and lens L1 in the tandem. A monochromator enclosed in a vacuum is used to determine sulfur, phosphorus and boron in steel in the vacuum ultraviolet in the range of wavelength of 100 nanometer region of the spectrum. Although the conventional ultraviolet region of the spectrum usually provides the best analytical lines for phosphorus and boron, common components of the steel interfere spectrally with both of these elements. AES can be used in the following: analysis of ferrous and non-ferrous alloys, determination of trace metal impurities in the alloys, metals, reagents and solvents, analysis of metals in geological, environmental and biological materials. Water analysis ICP instruments have been coupled with mass spectrometers to provide powerful analytical technique. AES is used in determination of gunshot wounds by the presence of metallic residues of GSR and bullets. ICP AES is used in the determination of species from the bone fragments. Atomic emission spectroscopy has long been a standard method for metal analysis. In atomic emission spectroscopy, a minute part of the sample is vaporized and thermally excited to the point of atomic emission. The energy required for these processes is supplied by an electric arc or spark, or more recently by laser or plasma composed of inert gas. It is convenient to distinguish these sources from the chemical flame sources used to obtain emission spectra. Instrumentation for AES may be divided into three major components: the sampling device and the source, the spectrometer and the detector, and readout device. AES can be used in the analysis of geological, environmental and biological materials, and also water analysis. ICP instruments have been coupled with the mass spectrometers to provide a powerful analytical technique. With all these informations here, we come to an end of today's lecture. Do keep in mind that we discussed today. I will be back with one more lecture in this series. If you want to learn more and enhance your knowledge, you may log on to our website www.cec.nic.in for MCQ, quizzes, LORs, etc. Thank you for your time today. I will see you in the next lecture. Have a great day.